So once upon a time, you learned what a connective tissue was in A&P1. And you know that a connective tissue is cells in a non-living extracellular matrix. So because of that definition, blood is a connective tissue because it is cells in a non-living extracellular matrix. In the world of blood, the cells become the formed elements, and the non-living extracellular matrix becomes the plasma. In lecture, we focused on all of these content areas: the characteristics of blood, a general pattern for circulation, for now, processes and regulation of those processes, and we actually emphasized in lecture and lab the formed elements and the plasma. For the sake of this discussion, we're going to choose some of those topics to review. So when we looked at functions of blood, you can divide them into 3 categories. And the categories are distribution, regulation, or protection. Distributive properties and functions of blood mean that you're moving or transporting something through the human body. And here you see a list of examples of things that would fall under distribution. For regulatory functions, you're dealing with the modification or the change in something. So blood is used to help us regulate body temperature. Because blood is little over half plasma, and plasma is mostly water, blood actually carries heat energy. So when we're hot, we carry the blood to our skin to release that energy. But when we're cold, we shunt blood out of our skin toward our core to conserve energy. We can also use blood to regulate pH, volume, and osmolarity of the body. And the last functional category was protection. Protection means you're responding to damage. So if you damage a blood vessel and you prevent blood loss, that's a protective function. And if you are invaded by any kind of pathogen, that's also a protective function. Now, component-wise, we know that, again, blood is a connective tissue. So it is cells in a non-living extracellular matrix. We'll talk about the cells in a little bit, but we can also briefly take a look at plasma. Plasma is 90% water. The vast majority of plasma then obviously is water. And dissolved into that water is solutes or molecules. The solutes contain proteins, lactic acid, urea, and creatinine, which are waste products, nutrient molecules, and the respiratory gases, which are oxygen and carbon dioxide. We're going to come back to proteins in a little bit and look at the different categories of proteins. You should be able to list some nutrient molecules off the top of your head. Lactic acid is a byproduct of muscular metabolism. Urea is a byproduct of breaking down proteins in the body or recycling proteins. Creatinine is a molecule that's an indication of kidney function. In other words, there shouldn't be tons and tons of creatinine in blood. If there is, it's usually an indication that the kidney is having some kind of pathology. So as mentioned, we're going to take a brief look at some of the more common proteins found in the plasma. Over half, nearly 60% of those floating proteins are albumins. Albumins serve to transport lipid molecules. In lecture, I talked about why. So if you don't remember why you can't just dissolve a lipid into the plasma, you should review that. The other common category after albumins becomes globulins. Their name kind of means really, really big folded up protein. And they serve 2 functions depending on the globulin. Some of them are for transport. Some of them are antibodies, and in that regard, they serve as protection. The third most common proteins you'll find in plasma function for blood clotting. And so, for example, we have 2 listed here, fibrinogen and prothrombin. Those 2 proteins are floating around in the plasma at all times. It's only when you have a trigger for the blood clotting process that these soluble proteins become insoluble and create a blood clot. Now, we can look at formed elements versus plasma in blood by running this test, looking at hematocrit. If you draw blood and put it in a tube, in the lab we talked about capillary tubes, very, very small tubes, and you spin it really fast, you separate it into its components. The dense red blood cells packed to the bottom, the kind of medium density leukocytes and platelets, or that thin layer in the middle, and then the most or least dense watery plasma stays on the top. On average, for normal males, hematocrit, or the percent of those red blood cells relative to the whole blood, is around 45%. Females, it's a little lower. The buffy coat, where we find the leukocytes and the platelets, that's less than 1%. There aren't a lot of those cells. So that means whatever's left over is plasma. So all these three together, erythrocyte or hematocrit, plus a little less than 1% for the buffy coat, if you subtract that off from 100, you'll find out what the plasma is. So the total of these three numbers should always equal 100. So let's take a look at our formed elements now. We have three categories: erythrocytes, formerly known as red blood cells; leukocytes, formerly known as white blood cells; and thrombocytes, or platelets. Erythrocytes in humans are anucleate. They have no nuclei. And in lecture, we talked about a couple of reasons why that might be. So you want to make sure you review that. We also discussed for a few minutes the importance of their shape, the fact that they are biconcave, or that they're kind of pinched in the middle. And once again, you should review the reasons why that's important. And pretty much everybody who's ever taken a biology class at all in any manner know that erythrocytes are for gas transport, oxygen. While they carry tons and tons and tons of oxygen, please realize they can also carry carbon dioxide. Now, because in humans and other animals, erythrocytes are anucleate, they're considered to be not really a true cell. On the other end of extremes, or the other end of our table here, we have our platelets, or our thrombocytes. They are responsible for the clotting mechanisms, and they're these tiny, tiny, tiny little purple dots here, smaller than a red blood cell. They are pieces of larger cells. The larger cells are called megakaryocytes. So if you make a megakaryocyte and you basically obliterate it into small little membrane-bound structures, you'll get a platelet. So once again, we have a cell here that's not really a cell because it doesn't have a full complement of organelles. The one population that does have a full complement of organelles are the leukocytes. So they are the true complete cells of this group. There are two categories of them that we'll get to in a second, and they are primarily for surveillance and immunity. So our erythrocytes first. Erythrocytes, we talked about the shape a second ago and mostly in lecture. And part of the reason that shape is there, but not all, is to enable them to have a large surface area. Because these gases, this oxygen and carbon dioxide, are going to diffuse across the cell membrane. So the more membrane they have, the more gas can diffuse. Erythrocytes contain tons and tons of hemoglobin, hundreds of millions of molecules of hemoglobin in one cell. And each hemoglobin can bind 4 oxygens, and here's why. Hemoglobin consists of 4 individual protein-based molecules that interlock to function. And you can see there's 2 alphas and 2 betas. That's based on structure. Now this long pink to reddish tube looking thing, that's the protein, which are called globins. And in the middle, this disc here is not protein at all. It's made of a different type of molecule, and that's the heme. It's within each heme that you find an iron ion, and that iron has the ability to bind the oxygen. So now you see why a hemoglobin molecule can bind 4 oxygens, because it has 4 hemes. The binding of that oxygen is reversible. So it loads the oxygen at the lung, but it'll unload the oxygen at the tissue. Now a red blood cell, an erythrocyte, can carry carbon dioxide as well. And it is reversible. And the movement is in the opposite direction. It picks up CO2 at the tissue and cell and drops it off at the lung so that you can exhale it. The deal with CO2 though is CO2 does not really bind to the heme. It binds to the globin, and we'll learn later in the respiratory system, it's also kind of secretly, slyly sequestered inside that cell in a chemical reaction. The last thing we want to mention about them is that erythrocytes are anaerobic. Because they lack organelles, they are not metabolically active. So they only need to make a teeny little bit of ATP to go about their day. That's important because to make ATP requires oxygen. And we want an erythrocyte to carry oxygen, not use oxygen. So by making them anaerobic, we ensure that they're only going to use a small amount of the oxygen that they need to carry. So, one of the other interesting things about erythrocytes is that they only live 100 to 120 days because they are anucleate. They don't have the hardware to repair damage to their membranes. So they do have a limited lifespan, which means you're going to need to constantly replace them. Furthermore, should your oxygen needs change, you might need to produce even more. The process by which we produce red blood cells is called erythropoiesis. It requires erythropoietin as the hormonal signal. So erythropoietin is often noted as just EPO. The other thing we're going to need are lots and lots and lots of stores of iron. Our liver stores it using a molecule I mentioned in lecture, and we can transport it with a different type of protein. But we're going to need we're going to need iron to make the heme. We'll need the heme to make the hemoglobin. Now all of this activity requires building blocks. So we're going to need protein to make all that globulin and enzymes to run this machinery. And we need lipids and carbohydrates. Lipids and carbs are for energy, and lipids can also be used for cell membrane. We're also going to need some molecules that help our enzymes work, and that's vitamin B12 and folic acid. So let's take a look at how this is going to play out. EPO is going to be secreted by the kidney, travel through the blood to the red bone marrow, where it stimulates bone marrow to go through a process to create red blood cells. The iron stores, your liver can store it by binding it up to proteins called ferritin and hemosiderin. If it wants to transport, hint hint, transport the iron, it's going to use the protein transferrin. Now it turns out that this is going to be a negative feedback loop. And here's how we make red blood cells. Let's say you travel to another location where that's of different elevation, and it's high elevation, as I mentioned in lecture. So you're not breathing in as much oxygen as you need. So what happens is your tissues become hypoxic, okay? In other words, your tissues have low oxygen levels. Well, one of the organs that monitors and detects that is the kidney. So here's your kidney. When your kidney detects that the tissues are hypoxic, it produces erythropoietin. Erythropoietin is secreted into the blood supply, where its target organ is the red bone marrow. And it will act on a stem cell that we'll name in a moment and trigger erythropoiesis. You can't get erythropoiesis without erythropoietin. The end result of triggering that process is that you're going to increase your erythrocyte production and release those red blood cells into the blood. That will raise oxygen levels in the blood because you have the ability to carry more. And if you meet your need, you'll shut the system off until next time. You will be asked erythropoiesis questions in your assessment future. So make sure you pause and you go through every single step and you have a good solid understanding of this process. So now that you have the picture, the big picture of the organs and the hormones involved, we can look at the cellular picture. Remember, I did say erythropoiesis is triggered by EPo. What organ releases it? You should have known the answer to that question. And what condition stimulates its release? And again, you should have known the answer to that question. Its target, one more time, is the red bone marrow. And here is its target cell, the hemocytoblast. This is a stem cell found in the red bone marrow. As I mentioned in lecture, it is the great-great-great-great-great-great-grandfather of an erythrocyte. It also happens to be the great-great-great-great-great-grandfather of leukocytes. And excuse me, it is also the great-great-great-great-great-great-grandfather of platelets. So every single formed element in your blood originally came from the stem cell called the hemocytoblast. Now, how does the hemocytoblast know what to do? How does it know that it needs to become an erythrocyte or one of the leukocytes or a platelet? It's the hormone signal that you give to the hemocytoblast that will determine what it becomes. So in this instance, it's becoming an erythrocyte because we put erythropoietin on it. Now, once we dump EPo on it, it's going to start to undergo shape changes and DNA changes. At that point, it becomes a committed cell, and it's going to become an erythrocyte if everything works out well. And we call it a proerythroblast. Proerythroblasts continue on their development until they eventually become a normoblast. And you can see by the time we get a normoblast, we have a cell that's changed in appearance dramatically, but it has also made a lot of hemoglobin. Remember, this cell is going to kick out its machinery, its organelles, and its nucleus. Once it does that, it can never make another molecule again, really, not a big molecule. So it's going to have to make all the big enzyme proteins and structural proteins it needs now, and it's going to have to make all the hemoglobin it's going to need now. When it's done accumulating all of those molecules, it actually ejects or spits out its nucleus. You can see it right there. Once it ejects its nucleus, it becomes a reticulocyte. The reticulocyte gets released from the red bone marrow into the blood supply, and it eventually finishes its development and becomes the erythrocyte. So once again, you will be asked questions about this process in your near future. So you want to make sure you have a solid understanding of this change from hemocytoblast to erythrocyte, what the important names and steps were along the way, and the changes that the cell is going through. Now, it all has to be highly, highly, highly controlled. If you don't have enough red blood cells, you'll become anemic. And you should understand why that is an issue. If you have too many red blood cells, that condition is called polycythemia. And just imagine taking the same amount of blood in the same blood vessels and shoving in extra cells. Do you think this blood is going to remain unchanged, become thinner and thinner, or become thicker and thicker? So again, you should understand the issue with polycythemia and why we don't want that either. Now, in lecture I said, what gets created must be destroyed. One more time, because these red blood cells do not have machinery, they cannot repair any damage that occurs, and their lifespan can be relatively short. They live only 100 to 120 days. So eventually they find themselves in organs like the liver, the spleen, and the marrow, and they begin to break down because their membranes become kind of rigid and fragile and break. What happens is they'll be phagocytized by a macrophage. The macrophage will take that hemoglobin and split it into the component parts, globin and heme. And now we can talk about what happens to those components. Remember, globin was just a protein. So we can ship that over to our liver and we can just recycle that. We can break it down and recycle the amino acids. But heme was not a protein, so it requires more work. So the liver takes the heme and it breaks it apart. It stockpiles the iron with its molecules there, and it converts the rest of it into a molecule called bilirubin. At that point, the liver has two options with bilirubin. Some of it will use to make bile, and the bile gets stored in the gallbladder. The rest of that bilirubin is taken to the intestines for excretion. Along the way, bacteria act on it and convert it into stercobilin, and it's really stercobilin that's excreted in the feces. So you can see when we break down a red blood cell and we deal with that hemoglobin, as much as possible is going to be recycled, and the last little bit that we can't really recycle well gets excreted. Just like with everything else, you should have a good understanding of this process and all the molecules along the way. So that was a lot about erythrocytes, but how about leukocytes? Remember, leukocytes fall into two categories. We have granulocytes, which means they have granules in their cytoplasm, and we have agranulocytes, which do not have granules in their cytoplasm. There are three granulocytes: eosinophils, basophils, and neutrophils. And there are two more or less agranulocytes, monocytes and lymphocytes. Do know there's a lot of different kinds of lymphocytes, but we'll get back to them in the immune system. So let's take a look at these guys and what they do. Neutrophils have very pale lilac colored granules that you can almost not see. They contain a lot of enzymes and molecules called defensins, and that makes neutrophil really good at destroying acute bacterial infections. They're often called bacteria slayers. They are most easily identified by this crazy pinched multilobe nucleus. Eosinophils have more of a U-shaped nucleus, but what makes them so easy to recognize are these red granules inside of them. They're responsible for the allergic response in part, and they're weakly phagocytic to parasitic worms. Basophils have a U or S-shaped nucleus, but good luck finding it because the nucleus picks up the same stains as their granules. So they almost look like one big blob here. Their granules will end up staining large and purplish black. Basophils play a role in inflammation because they secrete histamine and heparin. Histamine stimulates vasodilation, makes blood vessels get larger in diameter, so more blood arrives at a site of an injury. It also makes blood vessels leaky, so that once the blood arrives, the white blood cells can get out of the blood vessel to the intercellular spaces. Heparin prevents blood from clotting. That makes sense because if you want to bring blood to the site of an injury, and you want the cells, the immune system cells, to be able to leave the blood vessel and kind of move through to interstitial spaces, you don't want the blood to clot. So heparin's job is to prevent clotting. Lymphocytes, so now we're in the agranulocyte world. They have a big roundish nucleus that's pretty dark, and you can always see at least a thin rim of cytoplasm around them. Remember earlier I said lots of different types. T cells, for now, are involved in the immune response. And the reason we're so simplifying that is because there's a lot of different T cells. There's a couple of kinds of B cells, but for now we're going to simplify them into antibody production. And our last group that we're going to mention are the monocytes. They're relatively large. Their nucleus, it can be C-shaped or U-shaped sometimes. They'll eventually become macrophage, and they're bacterial slayers too, but for chronic infections. They also have the ability to do something rather special, and that's called presenting an antigen. Well, for now, presenting an antigen is a way of notifying all these T and B cells up here that an infection is taking place. Only macrophage can do that. So you should take a second, pause the slide, see if you can identify each of these 5 leukocytes. So obviously these have to come from somewhere. Now the good news is you have the ability to make more fairly quickly if you need them. So whenever you have an infection, that's where you're going to increase a customized population. So if you have a parasitic worm infection, you're going to increase your eosinophil population. That's how they work. The cells that kind of control this process actually are other immune system cells, so macrophage and T cells. They'll act on that hemocytoblast by secreting interleukins or colony-stimulating factors. The CSFs here, they are the ones that tell the hemocytoblast what specific kind of cell it's supposed to become. So let's take a look at that process. Here it is. Here's our hemocytoblast at the very, very top. And you can see it's got 2 pathways that it goes down first. If it becomes a lymphoid stem cell, that will differentiate to a lymphoblast, and it becomes a lymphocyte. So that one's pretty straightforward. The other option is to become a myeloid stem cell. When the myeloid stem cell becomes a myeloblast, just like the lymphoblast, it's a committed cell. And we can follow its progression to where we eventually see it become the 3 separate granulocytes or the monocyte. So you can see it's a little bit more complicated, but mostly because we're trying to make so many more cells. And just like with the erythrocyte production, if I circled it, you should know it. Right, lastly, we have our thrombocytes, or our platelets. They came from big cells called megakaryocytes. So we're going to make a megakaryocyte and then break it into pieces, basically. Thrombocytes, or platelets, are full of granules. And the granules contain serotonin, which is a neurotransmitter, but in this case, it's going to help us affect blood vessels. Calcium ions, lots of enzymes, and a molecule called platelet-derived growth factor. Based on its name, what do you think it stimulates? The fact that I'm circling the word growth is a big hint. Now, some of these molecules work together to form something called a platelet plug, which is part of a blood clot. And we'll go through the clotting cascade in more detail in lecture. We're just going to hint at it now. If you want to deactivate them or stop them from making the plug, you have to dump nitrous oxide on them. So now we've talked about the plasma, we've talked about the formed elements, and we looked at how we make these formed elements. So now let's talk about what happens if we damage a vessel. When you form a clot, that's only part of a process called hemostasis. Hemostasis is how you stop bleeding when you damage a vessel. And again, we've got 3 parts to it. The first thing we have to do is something called vascular spasm. If you cut through a little blood vessel, if it's an arterial, that blood vessel will respond to the damage by constricting to close itself off. That's a positive feedback loop, and it's in an attempt to close that vessel off at the site of the damage so that you stop losing blood. The second step is the platelet plug formation. That's when platelets who are in the vicinity chemically notice that there's a damaged vessel. And they'll stick to the walls of that vessel and stick onto each other and start secreting their granules. The serotonin and the calcium and enzymes in the granules interact with the proteins floating in the blood, and we start a cascade that leads to coagulation. Coagulation means that proteins come out of solution. It's not agglutination. We talked about that. That's different. That's from lab. That's antibodies and antigens. Coagulation comes about because we're going to convert proteins from a soluble form to an insoluble form. So here's the nutshell of these three. Vascular spasm is stimulated because of endothelial cells. These are cells that line the inside wall of the blood vessels. And if you slice through them, they release their cytoplasm, and there's different chemicals in their cytoplasm that will trigger the muscle cells of that blood vessel to contract. And these muscle cells are smooth muscle. It is also a positive feedback loop. It takes about 30 minutes for it to complete. In the platelet plug, remember we said if you slice through the vessel or you damage the endothelial cells. So the damaged endothelial cells are a trigger to platelets. Also, platelets are triggered by the presence of collagen, which they'll see inside the blood vessel if it's damaged. And unfortunately, a plaque, a rough surface like a fatty plaque, will also trigger a platelet plug formation without a damaged vessel. In any case, when platelets come in contact with the endothelial cell damage or collagen or plaque, they'll release serotonin. Serotonin stimulates chemotaxis. And this is how that works. Serotonin kind of acts like a stimulus to leukocytes in the area. So leukocytes that are actually quite far away from that area, not anywhere near the vicinity, will detect the serotonin at the site of the damage, and they'll begin to move toward that damaged vessel. In the meantime, the platelets are beginning to stick to the walls of the vessel and to stick to each other. Now, the other chemicals they secreted stimulated a cascade. And we'll talk in lecture about intrinsic versus extrinsic forms of that. But what happens in the cascade is you end up making this molecule, prothrombin activator. Prothrombin activator acts on the prothrombin already floating around in your blood and converts it to thrombin. Thrombin then acts on the fibrinogen floating around in your blood and converts it to fibrin. Fibrin is insoluble. It will come out of solution almost like little bits of spider web, and it will lace over the sticky platelets, and it will trap more platelets and more cells, and that's the point at which you get a blood clot. The good thing about a blood clot is that's the point at which you stop losing blood through that damaged vessel. And the blood clot becomes a trigger for repair. So take a second, pause on this slide, work your way through. I want you to focus on vascular spasm, platelet plug formation, and here, formation of the prothrombin activator to fibrin. And how that results in coagulation. This diagram, you can see in our red oval here, we have that part of the cascade that we're most concerned about learning. But you can also see here that it's illustrating intrinsic and extrinsic pathways. The end result of the pathways are the same. You get to the red circle. But the way you can get to the pathway differs depending on what the stimulus is. The intrinsic pathway deals with rupturing the vesicle and exposing collagen and things like that. The extrinsic pathway is just stimulated by tissues themselves, damaged tissues that secrete molecules that trigger the cascade. Now, not all clots stay in place. Once you start to heal, we're going to eventually get rid of this clot. And furthermore, before we even get to the healing process, we want to stabilize the clot. So what happens is, once a blood clot forms, it kind of squeezes on itself. And then that squeeze starts to trigger the repair process. The squeeze, or the retraction, squeezes extra serum from the fibrin and stabilizes the clot. It makes it stronger. The repair process gets triggered by that platelet-derived growth factor. That stimulates the rebuilding of the blood vessel wall. We bring in new fibroblasts that replace the connective tissue, and PDGF and other growth factors, will stimulate the endothelial cells to divide. This section right here, this concept here, was first presented to you in AMP1. At the end of the chapters on tissues, you should have learned about tissue repair. And it's obvious. Why do we want to control clotting? Well, if you can't form a blood clot, you're going to bleed. And if you form blood clots far too easily, you're going to block flow to a critical organ. That's actually what happens in some types of strokes, and it's definitely what's happening in a heart attack. In a heart attack, a spontaneous clot forms in the arterioles that bring blood to the muscles of the heart, and then those muscles spasm and die. So, how do we make sure we don't keep making clots forever? Well, eventually we start pulling clotting factors away from the site of the damage. Some clot is great, too much clot is bad. So we just remove the clotting factors and the clot doesn't grow or get any bigger. The other thing is we can have some leukocytes secrete inhibiting factors. So for example, fibrin kind of feeds back on thrombin and blocks it. So the moment you get tons and tons of fibrin around, you kind of stop activating the thrombin. Heparin actually interferes with part of the pathway above prothrombin activator. And we have this molecule antithrombin 3, which also inhibits thrombin production. So there are molecules that are released that inhibit the cascade so that it doesn't get out of control. Naturally in the body and unnaturally outside of the body, we do have mechanisms to try to prevent clots from forming spontaneously. Remember, a spontaneous clot in the blood vessels feeding the heart wall will cause a heart attack. So one of the things is that the inside lining of blood vessels and the heart is incredibly, incredibly smooth. That lining is called the endothelium. And when we get to blood vessel structure and the structure of the heart, we'll come back to this concept. But this is a simple squamous epithelia. The cells are tightly overlapping, and so it makes a smooth, friction-free surface. Also, endothelial cells have the ability to secrete a teeny little bit of heparin if they need to, so that'll in some cases help prevent a blood clot from forming. But let's just say you have a patient in the future that no matter what you do or they do, their blood is prone to clotting. And so these molecules can be given. Vitamin E quinone is an anticoagulant. Heparin obviously is. That's been purified long ago and can be administered or put into vacutainers to keep blood in its liquid form. Aspirin of all things. So if you've heard that people at risk of recurrent heart attacks are told sometimes by their doctor to take a baby aspirin, that would be why. It's to prevent that blood clot from forming in the coronary circuit. And another more powerful one, warfarin. So sometimes you hear people who are prescribed warfarin, and now you know why. Now, I'm not going to go through it because I went through it in lab and it takes an awful long time. But make sure you review blood typing and how to type blood for any blood type, what their antigens are, what antibodies they can produce, and how to make sure blood types are compatible. You will be asked in lab and lecture to figure out compatible blood types. If you've forgotten already, you can use this chart to figure out antigen and antibody for the blood types. You can review our rules for compatibility. Remember, you can always give blood to someone who matches. You can always receive blood from your match or ONEG, and then whatever else is compatible. Remember, to figure out compatibility, we have antibodies against antigens we don't have. So you need to focus on antigens and make sure that when you're matching recipient to donor blood, that the antigens are compatible. Also remember, a positive can receive a negative, but a negative can never receive a positive. That relates to Rh factor. And remember that the A The B and the RH are separate genes. They are inherited independently of each other. You should also understand and know why O neg is considered the universal donor and why AB positive is the universal recipient. Now we'll talk more about this in lecture when we do a bit more of a review on this. But there's one case where you can get an accidental combination in the wrong direction. So we want to avoid transfusion reactions, but the other thing we want to avoid is hemolytic disease of the newborn. That occurs when you have a mom who's Rh negative, but the baby could be Rh positive. And remember, a negative can never receive a positive. So what would happen if it's not treated is late in the pregnancy, the mom's immune system will attack and kill the baby and lyse the baby's blood. So we need to have a way of dealing with this. And the thing we use is something called RhoGAM. So I think it's around the 7th month of pregnancy. It might be before that, so don't quote me on the month. Mom goes in and she gets an injection of RhoGAM. And RhoGAM kind of makes mom artificially Rh positive until the baby's born. And then that way, she won't attack the baby. All right, so test yourself. Ms. Smith is blood type A positive. You should be able to figure out antigens, antibodies, and you should also be able to figure out who she can donate to and who she can receive from. Mr. Jones is AB negative. Same thing. And baby Mary is O positive. Same thing. Once you're done practicing, you should click on that link. So play the PowerPoint, click this external link. It's the blood typing game. If it doesn't work, the link breaks, just go to Google and type in blood typing game. This is a great game created by the Nobel Prize organization that will allow you to play with drawing blood, typing blood, and matching blood for patients in an emergency room. It's obviously all fake, so you can practice to your heart's content, and it's great fun. And that's it.